Hi, my name is Lily, and this podcast will examine the manufacturing and purification of dihydroxyacetone. Dihydroxyacetone is a bioproduct of great importance across a variety of industries. With the chemical formula C3H6O3, DHA is a triose carbohydrate which has significance in biological mechanisms of both eukaryotic and prokaryotic organisms. The molecule is produced as a byproduct of fructose metabolism in humans and oxidized glycerol in microorganisms. DHA has great industrial significance, and uses have increased since it was discovered to have tanning properties in the late 20th century. When the aqueous chemical was accidentally exposed to skin, scientists discovered it was capable of a browning action known as the Maillard reaction. DHA reacts with terminal amino groups in the stratum corneum in a reaction which produces pigments called melanoidins, resulting in a tanned appearance. DHA is therefore the primary active ingredient in almost all commercially available self-tanning agents and is widely considered to be very safe due to a lack of systemic absorption. Aside from the cosmetics industry, DHA is used in the pharmaceuticals industry and was originally developed as an alternate source of glucose for diabetic patients in the 1950s. The food and wine industry uses of DHA mainly include flavouring and colouring of products. The compound can be obtained from plant sources such as beets and sugarcane or chemically synthesized. However, for the most economical method is by fermentation of glycerin. Glycerin is the main byproduct of biodiesel production, an industry which is growing in size significantly, and due to this, the cost of glycerin has plummeted. DHA prices are between 250 and 500 times higher than glycerin alone, and therefore this relatively cheap waste product is an ideal candidate for value adding, generating huge profit margins for companies who produce it. One method of producing DHA is through biological oxidation. Acetic acid bacteria are commonly used as biological producers, and once placed under environmental stress, can be utilized to produce vast quantities of DHA. Production of DHA by microorganisms occurs by incomplete oxidation of glycerol. This process is catalyzed by glycerol dehydrogenase, an enzyme bound within the cytoplasmic membrane of acetic acid bacteria, which is responsible for glycerol metabolism in many aerobic bacteria. After completing a literature review of the biological methods of DHA production, the following procedure has demonstrated a 94% efficiency of glycerol oxidation, and in laboratory models, a DHA concentration of 27.2 grams per litre was observed after a 5-day incubation. The strain of bacteria shown to most efficiently produce commercial quantities of DHA is Glucanobacter Oxydans. An obligate aerobe, G. oxidans uses glycerol as an energy source and through oxidative fermentation produces DHA as a metabolic byproduct. This bacterial strain can be genetically modified to overexpress glycerin dehydrogenase in order to metabolize larger quantities of glycerol. The main drawback of this bacterial strain is that when high concentrations of glycerol are present in aqueous culture medium, oxidans growth is inhibited and therefore subsequent DHA synthesis. Glucanobacter oxydans can easily be obtained from a laboratory, and a selection for a single strain on agar is straightforward. After the desired strain has been isolated and identified, a starting culture can be maintained on a sorbitol agar dish containing 50 grams per litre sorbitol and 20 grams per litre agar, stored at 4 degrees Celsius. The bacterial strain is then passaged every 30 days onto fresh culture medium, containing the same components. This culture is incubated at 28 degrees Celsius for at least 24 hours prior to transferal to a fridge for storage. For biomass proliferation, pure culture is transferred to a liquid medium containing 30 grams per litre yeast extract and 20 cubic centimetres per litre of ethyl alcohol, and kept at a pH of 5, incubated for 24 hours at 28 degrees Celsius with an impeller speed of 700 rpm. In order to activate glycerin dehydrogenase, 1% volume per volume of culture is transferred to an activating medium in a bioreactor containing 5 grams per litre yeast extract, 20 grams per litre glycerol, and 5 grams per litre ammonium sulfate. kept at pH 5 for 28 degrees Celsius for 48 hours with an impeller speed of 700 rpm. After glycerin dehydrogenase has been activated, 5% volume per volume of free cell inoculum is transferred to a bioreactor containing 30 grams per litre sorbitol, 5 grams per litre yeast extract, 0.5 grams per litre magnesium sulfate, 1 gram per litre dipotassium phosphate, 2 grams per litre calcium carbonate, and approximately 30 grams per litre of glycerol to begin the process of biotransformation of glycerol to DHA. Proportions of glycerol must be carefully monitored as an oversaturation of glycerol inhibits cell growth. Fermentation medium is then cultured for 24 hours with 220 rpm stirrer speed at 30 degrees Celsius. Glycerol is most efficiently biotransformed during the log phase of growth, and the pH of culture medium is kept at 5 until fermentation is due to begin, at which sodium hydroxide is added to increase the pH to 6. To control foaming within the bioreactor, polyether ketone is added as needed. Fermentation is most effective when compressed oxygen supply is steeled and stirrer tank reactor is utilised, as oxygen is the terminal electron acceptor in the fermentation process, which allows for the production of DHA. Crucially, glycerol and DHA concentrations must be monitored within culture medium for the duration of the fermentation process. To measure glycerol concentrations, enzymatic glycerol reagent test kits are used. The test kit contains a complex of 3 enzymes catalyzing 3 reactions which include glycerol phosphorylation, glycerol 1-phosphate oxidation, and quinonomine, which produces a pink-purple colour and can be measured on a spectrophotometer at 540 nanometers to determine glycerol based on optical density. The concentration of DHA can be measured by gas chromatography or spectrophotometry. For spectrophotometric analysis, a 3 cubic centimetre sample of inoculum is combined with an equal volume of 2,3-dinitrosalicylic acid, which is incubated at 100 degrees Celsius for 10 minutes. The sample can then be measured at 550 nanometers to determine the concentration of DHA present. Throughout literature, the highest feasible concentration of DHA achieved ranges between 25 and 28 grams per litre of pure DHA. After sufficient quantities of DHA are synthesized from glycerin, the compound must be released from its intracellular storage. For this, the ultrasound method can be used to lyse cells. Sonication at 210 watts and 18 kHz for 5 minutes is enough to release DHA produced and prepare the culture broth for downstream processing and purification. Downstream processing and purification of dihydroxyacetone. Once liquid fermentation broth is saturated with DHA and cells are no longer metabolizing glycerin, the products must be separated, purified, and prepared for their distribution to various industries, complying with any regulations that may be necessary. Downstream processing accounts for almost 50% of costs in many biotechnological processes, and as a result, the methodology used must be carefully examined and optimized. Though I was unable to ascertain a precise sequence of downstream processes for DHA from companies, the following techniques are used on a variety of similar compounds which have undergone oxidation from glycerol biologically. Each methodology presents individual challenges, and it is likely that a combination would yield the best results. Solid-liquid separation. Techniques that are commonly used to separate inoculum and aqueous compounds from cellular debris include microfiltration, centrifugation, and decanting. Flocculation precipitation, however, is the most effective large-scale separation technique. Flocculation is a technique which utilizes the chemical properties of cells within a culture to bind to an added floc to increase the size of particles, making filtration and separation of cellular substances from culture medium simpler and more accurate. This phase of processing removes cellular debris from the culture containing the aqueous desired compounds. Evaporation and distillation. Evaporation and distillation are commonly used in the biotechnology industry to remove water and other unwanted molecules from a broth. Multi-stage evaporation or down-film vacuum evaporation are the most desirable methods as they save energy as compared to single-stage evaporations. In down-film vacuum evaporation, 2 vacuum rectification columns are used to remove water and acids and recover the desired compound respectively. Prior to evaporation, desalination is carried out by electrodialysis. If salt is not removed from the slurry, a lower rate of evaporation occurs, with a lower yield of final product. Soluble proteins are additionally precipitated by addition of alcohol into concentrated broth after evaporation. Methanol has been determined as the safest alcohol that can be utilized for crystallization, as it complies with global environmental and health standards and can be purchased for $1.50 per litre, which is comparatively cheap. The main problem with this method is the high cost, and desalination is required prior to the process, driving prices up higher. Membrane separation. Many similar compounds to DHA utilize multiple membrane types for fine filtration. Initially, microfiltration, then ultrafiltration, and finally nanofiltration remove all particles greater than 0.2 micrometers and molecules with a weight greater than roughly 200 to 400 daltons. The power consumption for the filters are quite high, however the process is quite effective at removal of desired compounds. After initial filtration, another rough evaporation aids in removing excess water. X-type zeolite membranes have been used to separate compounds similar to DHA from their precursors, glycerol, and any other carbohydrates present in the resulting medium. Zeolite membranes are chemically produced approximately 250 nanometer wide membranes which contain charged silicon or other ions to separate compounds based on physicochemical properties. The exact membrane to be used for this stage is unclear, and the chemical properties of DHA would need to be examined in order to determine the type of zeolite filter, as they can be enriched and modified based on requirements for separation. Chromatography. In similar compounds to DHA, a strong cation exchange resin of polystyrene sulfonate has been used to separate the desired products from impurities. In laboratory models, original feed solution was diluted up to 10 times with water in the process of liquid chromatography. However, experiments demonstrated purity of products obtained as more than 95%. The main downside of liquid chromatography is the extensive dilution, and a further stage of evaporation would be required should this method be applied. Liquid-liquid extraction. Liquid-liquid extraction has the benefit of large throughput and lower energy consumption than distillation. Examples of this extraction technique utilizing organic solvents have been applied to similar organic molecules to DHA. The process may be able to extract the product from a dilute broth if a suitable solvent is found. Based on research, it appears methanol may be an effective solvent for DHA extraction. However, this is only speculation and more research into the methodology would be required. Crystallization. Finally, crystallization has been shown to be a promising method of extraction, with recent studies finding it highly effective in precipitation of DHA. Solvent and crudely separated aqueous product must be heated to between 10 Celsius and 30 degrees Celsius and left to homogenize with magnetic stirring. After between 96 and 288 hours, the resulting precipitation can be drawn back through a filter to separate crystals of DHA from liquid. Crystallization results in a highly purified product. This technique would likely be the most effective when combined with initial membrane separation and potentially an ethanol salt distillation process further down the line. After DHA has been purified and crystallized, it's ready for distribution. In summary, dihydroxyacetone is a highly worthwhile biotechnology product to produce, and with demand for the compound increasing, it would prove to be a highly lucrative investment to make. Thank you for your attention.